Sometimes the absolute fastest way to destroy an ecosystem is to, like, try and save it.
Oh, absolutely. It happens way more often than you'd think.
Right. And today we're actually looking at a case study where a caretaker tried to give their outdoor pond just a routine health boost, and they accidentally triggered this total microscopic apocalypse.
A complete system collapse.
Exactly. So welcome to this deep dive. You and I are going to play ecological detectives today. We're going to unpack this really fascinating set of fish-keeping notes that document the sudden, catastrophic collapse of a miniature aquatic ecosystem.
And we're diving straight into the mechanics of that failure, right?
Yeah.
Looking at how a super common, highly accessible water treatment basically acts as a triple threat ecosystem destroyer when it's used the wrong way.
Yeah, and we'll get into the fundamental biology behind why it happens, which is just mind-blowing. But let's set the scene first. What are we actually looking at here?
So the notes center around these outdoor ponds, right? And the primary plant life they're cultivating is called Common Salvinia. And for a long time, the system was doing great,
Yeah.
highly successful. The Salvinia is, it's a floating fern, and it had formed this thick, super healthy canopy across the surface of the water.
So it was operating exactly as you'd expect a self-regulating outdoor setup to work.
Exactly. It was thriving.
But then the notes document this super localized anomaly. Like suddenly, in these very specific spots across the ponds, the salvinia didn't just slow down, it turned brown and the growth completely stalled out.
Yeah, it created these like isolated dead zones right in the middle of this otherwise lush canopy.
Which is so weird. It's like having a vibrant, bustling neighborhood where, I don't know, one specific street suddenly becomes a total ghost town.
That's a really good way to put it.
And the caretaker's first response, naturally, was to take healthy salvinia from a neighboring pond that was doing fine and transplant it directly into these failing spots to, like, jumpstart the biological activity.
Right, which makes sense on paper. You see a dead patch, you plant new stuff.
But wait, if we're dealing with a localized dead zone in an outdoor pond, I mean, a pond is essentially an open system, right? It's exposed to the same sun, the same rain, the same temperature across its entire surface.
Exactly, it's all the same environment.
So transplanting healthy plants into that spot seems like moving new tenants into a building that has fundamentally broken plumbing. Why wouldn't transplanting healthy plants just fix the issue?
Well, because the environment itself had actually been fundamentally altered just days before.
Oh, really?
Yeah, the ecosystem didn't just randomly fail, it was pushed. The notes reveal this critical shift in the maintenance routine. Just before these dead zones popped up, the caretaker performed some significant water changes.
Okay, water changes are normal.
Right. But during that process, they added a mixture of standard aquarium salt and a chemical called methylene blue.
Wait, methylene blue?
Yep.
I mean, we all know methylene blue is this very traditional, widely used medicinal dye in the hobby. You see it heavily utilized for, like, treating fungal infections on fish eggs or dealing with external parasites.
It's an absolute staple in quarantine tanks.
Right. But how does introducing a standard, heavily diluted medicinal dye into a massive outdoor pond trigger a botanical collapse? Like, why would that kill the plants?
It actually comes down to the physics of light penetration.
The physics of light.
Yeah. Because methylene blue is this intensely potent synthetic dye. Even when it's highly diluted, it significantly alters the optical properties of the water column. I mean, it literally turns the water a deep, impenetrable blue.
Okay, I can picture that. But what does that do to the plants?
Well, we have to look at how a plant like Salvinia actually processes light, right? Photosynthesis relies heavily on what's called photosynthetically active radiation, or PAR.
Right, the TR spectrum.
And the most critical wavelengths for chlorophyll absorption are in the red and blue spectrums.
Wait, so if the water is dyed blue, that means the dye is reflecting blue light and absorbing all the other wavelengths.
Exactly.
So it's actively stripping the red spectrum out of the light that reaches the plant.
That is the core of the first failure right there. Water naturally filters out red wavelengths faster than other colors anyway, but when you introduce a heavy synthetic dye like methylene blue, you aggressively accelerate that absorption.
So you're basically starving it of red light.
Exactly. The dye acts as an artificial light filter. So the salvinia is sitting right there under the open sky in the broad daylight, but because of the altered refraction and absorption in the water right around its root-like structures, it is effectively starved of the specific photons it needs to excite the electrons in its chlorophyll.
Oh, wow. So by adding this treatment to the water, they essentially threw a massive blackout curtain over the pond.
Yep, completely block the light it needs.
It's like putting heavy sunglasses on a houseplant.
That's a fantastic analogy. Yeah, the biological engine that drives the plant simply loses its power source.
But, okay, if you drastically reduce the available red spectrum, I'd assume a floating plant like Salvinia would just, I don't know, stall out. Or maybe go dormant until the water clears up and the dye naturally breaks down.
You would think so, right?
Yeah, but the notes show they didn't just go dormant. They actively turned brown and literally rotted away. Why does a lack of light cause immediate cellular death instead of just a pause in growth?
Because the light starvation is only the first phase of the damage. This is where we move from an external physical block, the blackout curtain, as you said, to a devastating internal biochemical disruption.
Wait, it gets worse.
Oh yeah. The Salvinia isn't just passively sitting in that dyed water, like all vascular plants. It actively draws water and nutrients up through its modified fronds, which basically act like roots.
Oh no, so it's actively drinking the dye.
Yes, it draws it right up into its internal cellular structure. It's essentially a Trojan horse scenario.
That is terrifying.
Right. The plant's own biological imperative to hydrate becomes the exact delivery mechanism for its destruction. It pulls that methylene blue directly into its vascular tissue.
You know, we actually use methylene blue in microscopy as a biological stain specifically because it is incredibly effective at binding to cellular structures.
Exactly, especially nucleic acids.
So, what happens when a living, actively respiring plant cell suddenly becomes saturated with a synthetic redox dye?
It causes a devastating metabolic short circuit. You see, methylene blue is an electron carrier. In a lab setting, we use it to study cellular respiration because it can literally intercept electrons in the electron transport chain.
Okay, so inside the living cells of the Salvinia.
The dye effectively hijacks the plant's internal energy production. It physically interferes with the mitochondria and the chloroplasts, completely preventing the cell from producing ATP.
And ATP is the energy currency of the cell.
Exactly. Without ATP, the cell dies.
So the plant isn't just starving because it lacks light from the outside. It's starving because the methylene blue has bound to its internal cellular machinery and fundamentally broken its ability to process energy, even if the light was there.
Precisely. The plant's own vascular system is pumping a metabolic poison directly into its cells.
Wow. Which manifests visually as the plant literally rotting from the inside out.
Right. The cellular walls break down, necrosis sets in, and those floating fronds just turn brown and disintegrate.
So it's a dual front attack. The dye blocks the necessary light wavelengths from the outside and then simultaneously dismantles the cellular respiration process from the inside.
Exactly. It's brutal.
Ah.
And here is where it becomes a tree apocalypse. It doesn't stop with the plants.
Okay, let's talk about the biological filter then. Because we all know how vital the biofilter is for processing ammonia in any aquatic setup, right?
Oh, absolutely essential.
We completely rely on those nitrifying bacteria to keep the nitrogen cycle stable. But, I mean, I've always operated under the assumption that an established, really robust bacterial colony in a massive outdoor pond would be, somewhat insulated from a simple water conditioner or a standard medicinal dye.
A lot of people think that.
Yeah. So how does methylene blue physically wipe out an established biological safety net like that?
Well, think about why we use it. The mechanism of action that makes methylene blue so incredibly effective against fungal infections on fish eggs is the exact same mechanism that destroys the biofilter.
Because it's a broad-spectrum antimicrobial agent.
Exactly. It does not possess the chemical intelligence to differentiate between, say, a pathogenic fungus attacking a fish and the nitrosomonas and nitrobacter bacteria that are happily oxidizing toxic ammonia into nitrites and nitrates.
Oh, I see. Because bacteria also rely on cellular respiration. If the dye penetrates the bacterial cell walls and intercepts their electron transport chain just like it does in the plant cells, they lose their ability to produce ATP and they just die.
The disruption is instantaneous.
Yeah.
The dye causes massive oxidative stress within the bacterial cells. So that entire microscopic workforce, the millions of bacteria that make up the biological filtration system, is eradicated by the exact same metabolic short-circuiting that killed the Salvinia.
Okay, so if it's not just starving the plants of light, but it's a systemic poison that takes out the plants and the bacteria, it's like taking a sledgehammer to a delicate watch.
That's exactly what it is.
But if that biological safety net is suddenly just gone, wiped out, how quickly does the chemistry of that pond water actually turn lethal? Like, are we talking about a slow decline over a few weeks, or is the toxicity immediate?
In an outdoor pond, especially one exposed to sunlight and varying temperatures. The timeline is measured in hours.
Yeah.
Maybe a day or two at absolute most, depending on the bioload.
Just hours.
Yeah. Think about it. The decaying Salvinia alone is now contributing massive amounts of organic waste directly into the system.
Hmm.
And as that organic matter breaks down, it produces ammonia.
Right, and without the nitrosomonas bacteria there to consume that ammonia, it just accumulates rapidly.
It has nowhere to go.
And since this is an outdoor pond, temperature and pH play a huge role in ammonia toxicity. As the water warms up in the sun, the relatively harmless ionized ammonium shifts into highly toxic unionized ammonia, and the biological filter would normally process that before it ever becomes an issue, but the dye has neutralized the filter entirely.
Yep. The caretakers unintentionally engineered a cascading failure. This intervention that was designed to proactively treat the pond resulted in physical light starvation, internal biochemical poisoning of the primary plant life, and the complete chemical eradication of the nitrogen cycle.
Wow.
The environment went from completely self-regulating to highly toxic, purely due to a miscalculated chemical input.
Okay, so how do you even recover from that? The notes mention that following this collapse, the caretaker completely revised their maintenance protocol, right?
They did. They learned a very hard lesson.
They stopped using methylene blue as a general preventative entirely and switched exclusively to using regular, untreated aquarium salt for normal water preparation and routine changes.
Which is a much, much safer approach.
Now, we utilize salt constantly in the hobby to promote slime coat production or to treat minor ailments. But mechanically speaking, why is plain untreated salt the sustainable long-term choice here versus a broad-spectrum dye? What does the salt actually do?
It all comes down to osmotic regulation and biological compatibility.
Osmotic regulation.
Yeah. So freshwater fish are constantly expending energy to maintain their internal homeostasis. Because their internal fluids are naturally saltier than the surrounding pond water, water from the pond is continuously trying to diffuse into their bodies.
Through osmosis.
Right. So they have to constantly process and expel that excess water, which takes a pretty significant metabolic toll on the fish.
Oh, I see. So when you add a measured dose of untreated aquarium salt to the pond, you are slightly increasing the salinity of the surrounding water, which reduces that osmotic gradient between the fish and its environment.
Exactly. Which means the fish just don't have to work as hard to maintain their internal fluid balance.
Their osmoregulatory system gets a break.
Exactly. It reduces their overall stress, which naturally allows their immune systems to function way more efficiently.
So you are providing a systemic health benefit to the fish, but you're doing it without introducing a synthetic chemical that, alters light refraction, binds to plant DNA, or completely interrupts bacterial cellular respiration.
Right. The salt provides a gentle, supportive environmental tweak, whereas the methylene blue operates as a total scorched earth biological reset.
Which, to be fair, a scorched earth reset makes sense if you are, say, setting up a quarantine tank from scratch, and you really need to ensure a completely sterile environment before introducing a new, potentially infected fish.
Sure, in that specific scenario, it's very useful.
Because you don't have an established plant canopy or a mature biofilter to protect in a bare quarantine tank.
Exactly.
But deploying that level of chemical intervention as just a casual additive during a routine water change, it's treating a basic maintenance task like a level 4 biohazard emergency.
It is. It fundamentally misinterprets the very nature of a closed biological system. True expertise isn't just knowing what a chemical does, it's knowing when to use it.
It's the difference between taking your daily vitamins and taking a heavy-duty, broad-spectrum antibiotic. You don't take the antibiotic just because you sneezed, or you'll wipe out your entire gut microbiome.
That is exactly it. The strength of an ecosystem, whether it's a giant outdoor pond or a tiny 10-gallon desktop tank, lies in the complex, mostly unseen relationships between the light, the plants, the microorganisms, and the larger fauna.
Right.
When you introduce a heavy-handed chemical intervention like methylene blue, you aren't just treating a symptom, you are forcing a radical new variable into every single one of those complex relationships simultaneously.
Man, we have traced an incredible biological cascade today. We looked at how a standard medicinal dye, applied casually, acts as a literal physical light filter that deprives plants of the red wavelengths they need to synthesize energy. We broke down that terrifying Trojan horse effect, where a plant's own vascular drive to hydrate literally pulls a metabolic poison deep into its cellular structure, short-circuiting its ability to produce ATP and causing internal necrosis.
Rotting from the inside out.
Right. And then we mapped how that exact same chemical disruption completely eradicates the nitrifying bacteria of the biofilter, causing a rapid lethal spike in unionized ammonia.
The microbiome massacre.
Yeah. And finally, we looked at how returning to the fundamental mechanics of osmotic pressure, just using simple untreated aquarium salt, provides a stable, non-destructive path forward.
I think the overarching lesson documented in these notes is really about restraint. In any complex system, the absolute most powerful tool a caretaker has isn't a chemical treatment. It's an understanding of the underlying biological mechanisms at play.
Absolutely. Well, I want to leave you with a final thought to ponder as you look at the systems you manage in your own life, whether that's a complex project at work, maybe your personal fitness routines, or even just your daily habits. Where else might you be deploying a scorched earth, methylene blue level intervention to a situation that really only requires the gentle, balancing friction of a little untreated salt? How often do our well-intentioned, heavy-handed attempts to optimize a system end up actively dismantling the very foundation we're trying to build upon?
That's a great question to ask yourself.
Thank you for joining us on this deep dive today. Keep questioning the mechanics behind those quick fixes, respect the complex systems around you, and we'll catch you on the next one.